Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Application of Treadmill Exercise Using Mice as a Model for Studying the Influence of Maternal Exercise on Offspring Metabolic Reprogramming. My name is Darius Sulam from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Columbus Instruments, so a big thanks to them for helping to make this webinar possible. Our speaker today is Dr. Jun-Suk Sun, Assistant Professor in the Departments of Obstetrics, Gynecology, and Reproductive Sciences and Physiology at the University of Maryland. In just a few moments, Dr. Sun will be discussing his latest work on how a maternal exercise of mice protocol can aid in explorations of the beneficial effects of exercise on neonatal health. So now I'm going to welcome our presenter, Dr. Jun Sun. Jun, thanks so much for being here.
Hi. Hi.
The floor is yours whenever you're ready.
Yeah, I'm ready, and I can start. So, hi. I am Jun-Suk Sun from the Department of Obstetrics, Gynecology, and Reproductive Sciences, and Department of Physiology of University of Maryland School of Medicine. First of all, thank you for watching and for having an opportunity to talk our research. So today, I'm gonna talk about the beneficial effects of exercise training during pregnancy on offspring metabolic health, as well as I'm going to introduce the treadmill exercise protocol as an exercise model for pregnant mice. Before we begin, I would like to inform you guys that today's presentation will focus a little more on the data available through the treadmill exercise model. Okay, let's start. There are a number of environmental factors that can be influential to reprogram fetus. As one of them, high-calorie diet may induce maternal obesity, which deteriorates fetal and infant metabolic homeostasis. And when the baby grows up, they easily become obese and suffer from metabolic disorders such as type 2 diabetes and cardiovascular diseases. However, the exercise, which has lots of benefits for mother and her baby, can protect the negative impacts caused by maternal obesity. This also has a long-lasting effect in metabolic homeostasis when they grow up. So my lab aims to figure out what is really going on during metabolic adaptation or mode adaptation at the molecular level or cellular or system levels. And way I look at it in this study, we really wanted to understand what's going on at the molecular levels in response to exercise and pregnancy. So, how does exercise during pregnancy prevent and keep their offspring healthy? We have tried to discover the reasons from fetal metabolic reprogramming with emphasis on brown fat and muscle. So, for the last several years, we are trying to gain fundamental understanding for brown fat. This is a really great model for investigating mitochondrial biology because it contains dense mitochondria in the mammalian cells, and one of the functions of brown fat is energy consuming very well through thermogenesis, which I'm gonna talk more in the moment. This feature is completely opposite from white adipose tissue, which has low density of mitochondria, and it is essential for energy storage. High density of mitochondria in brown fat generates most of chemical energy required for cells' biochemical reactions. Glucose and triacylglycerol are utilized as sources of energy through glycolysis or lipolysis and beta oxidation. So we can say that mitochondria are the power stations making energy. The major function of brown fat is thermogenesis, which is the process for generating heat by mitochondrial protein carrier called uncoupling protein 1, UCP1. And UCP1 localizes to the mitochondrial inner membrane, and it generates heat by dissipating the energy proton gradient from the electron transport chain in mitochondrial respiration. UCP1 is the only thermogenic protein for the past years, as we mentioned, and however, it is recently reported that beige fat thermogenesis occurs in UCP1 independently. In the skeletal muscle, sarcolipin and SERCA, SERCA, which is sarcoplasmic endoplasmic reticulum calcium transport ATPase, and SERCA-dependent non-sugaring thermogenesis is dominant in the skeletal muscle. Similar to the muscle-based thermogenesis, subcutaneous beige fat we mentioned has a SERCA-dependent thermogenesis as well, which is a little bit different with muscle thermogenesis, but which is a new concept that distinguishes it from the original UCP1-dependent manner. As we talked about UCP1-dependent or independent thermogenesis for the last long time, all about thermogenic signaling is by UCP1-dependent. But clearly, as we mentioned, the UCP1-independent pathways exist. So there are multiple pathways to generate heat in brown and beige fat. And I'm going to more emphasize that brown and beige fat secrete a number of molecules, such as peptides, lipids, microRNAs, and so on, which are called cytokines, arise from brown adipocytes. This is very important in tissue and system levels. And the last part is the metabolic role of brown and beige fat. In the previous slide, I showed the mitochondria as power stations, and glucose and fatty acid are utilized to generate ATP as a form of energy. These features in brown and beige fat locally and systemically regulate glucose and insulin metabolism. Thus, developing and growing brown and beige fat are critically important for metabolic homeostasis. In particular interest for us regarding UCP1-dependent mechanisms, there are a number of previous studies that exercise training enhances the functional role of brown fat and its thermogenesis. Furthermore, in adults, subcutaneous white adipose tissue can be adapted to brown adipose phenotype, like this. And this process of browning white adipose tissue is accelerated by physical activity. This is very important point, and we call them beige or bright adipocytes. These are really similar with brown adipocytes in terms of high mitochondrial density and energy dissipation and thermogenesis. So, Why and how can physical activity stimulate tissue development and adaptation? In terms of gene and molecular regulations, acute exercise dramatically increases gene expression, but uh, its half-life is relatively very short. However, regular and chronic exercise training gradually increases the baseline of gene expression and its protein levels, as we shown in this slide. Uh, the good thing in terms of chronic exercise induced elevated baseline of gene and protein levels is that the target molecules are continuously produced even we are not going to do exercise. Uh, health benefits of physical activity is closely associated with the change of baseline of molecules, as I mentioned. And what I'm talking about is that acute exercise only has acute effect of exercise. On the other hand, chronic and regular exercise can have long-term effects of exercise, which include brain health, heart health, cancer prevention, and bone health. Exercise training is even also impactful for pregnant women. Um, Many studies in recent systemic and meta-analysis show that people with physically active lifestyle during pregnancy, no matter maternal weights, such as normal weight or obese weight, have advantages in children, such as birth weight management, prevention of preterm birth, and children obesity. These studies clearly show that these benefits include the offspring of women who are healthy and in shape. So whether pregnant women are healthy or obese, working out is truly beneficial. And today, I'm going to introduce a great exercise model to investigate the beneficial effects of exercise during pregnancy on placenta and fetal tissues, including the brown fat and muscle. In the animal studies, there are two representative exercise models commonly used in mouse studies. The first is a voluntary field running exercise program, and the second is forced treadmill exercise program. These two exercise methods have their own strengths and weaknesses, so I recommend the investigators can choose according to their purposes. So, to briefly show the pros and cons of each method, there are these points. In the first treadmill exercise program, we can control um, exercise duration and intensity exactly, which can reduce the variation of exercise volume between mice. However, treadmill exercise induces stress mentally and physically because it is the forced exercise, which means that they should exercise no matter they want to exercise or not. On the other hand, the pros and cons of hill running exercise model are totally opposite with forced exercise. Field running exercise has the advantage of not being stressed because mice can voluntarily exercise when they want. But the disadvantage is that they cannot match the amount and volume of exercise between mice exactly. So in our study, to show exact exercise intensity, we selected the treadmill exercise model based on our purposes. To figure out exact exercise protocol for pregnant mice, we used the proposed aerobic exercise guidelines for pregnant women published in Sports Medicine several years ago. to set the intensity of exercise. And we can find that they consider three different intensities based on the oxygen consumption rates, and considering that it is the first trimester up to 13 weeks and the second trimester up to 26 weeks and the third trimester up to the rest of the delivery. We are able to set the exercise program for pregnant mice largely in time for the 3 phase. And using oxygen consumption levels in setting the exercise intensity have been considered as a gold standard for aerobic exercise. We also measured oxygen consumption rates and used them to set the target intensities. Considering there are 3 trimesters, first, second, and third trimester for pregnant women, we also divided into 3 different stages: early stage, midterm, and late stage, to reduce the risk of miscarriage in the early stage of pregnancy. 40% of VO2 max, the maximum oxygen consumption rates, was applied in the early stage. And considering the increased body weight of maternal mice because of the weight of fetuses and their growth in the late stage, the exercise intensity was adjusted to 50% of VO2 max. And I want to show the video of mouse treadmill exercise. And we try to keep the mice as stress-free as possible. And instead of using the electric shock at the backside of the treadmill, we put the sponge in the backside of treadmill to encourage the mice to run well, like this. And if you guys need more information about our protocol, please see our protocol paper using this link. And please watch our short video. They can run very well, right? Okay, and following our unique treadmill exercise training, the Offspring had a high fat diet challenge for 8 weeks after weaning in order to induce obesity, which was performed to figure out the preventive effect of maternal exercise on offspring metabolism. And we could find that maternal exercise prevents the increase of body weight of female offspring, but not in male offspring, which was because of increased skeletal muscle, especially in males. We could also show the lipid droplets in the offspring liver through the oil residue staining, one of the technique, which stains the lipid red, like this. And we can also show that maternal exercise protects offspring to hepatic steatosis. Consistent with the offspring lipid profiles, maternal exercise also showed the protection of glucose intolerance caused by 8 weeks of high fat diet challenge, like this. So this study, together with other works by other groups, we can suggest that maternal exercise can control the offspring metabolic health. This is very exciting. And as an evidence how to control offspring metabolism in response to maternal exercise, we could show that maternal exercise enhances brown and beige thermogenesis, and we could show that maternal exercise increases surface temperature of interscapular region, this region of the backside. And it also protects against the increase of lipid droplet in the brown adipose tissue. Okay, and I want to more emphasize the exercise during pregnancy regulates mitochondrial activity-related pathways, which include inner-membrane activity, respiratory chain activity, NADH dehydrogenation, fatty acid oxidation, and beta oxidation. And these data are from transcriptome analysis using fetal brown adipose tissue. So far, we are saying that exercise in pregnancy develops the fetus and positively controls the metabolism of offspring. So, how does maternal exercise mediate fetal development? So, we firstly targeted the placenta as a mediator linking maternal exercise to fetal reprogramming because oxygen and nutrients from mother are transferred across the placenta to the fetus, and it is the only organ can support the fetus. Indeed, recently there are a number of reports that placenta secretes molecules such as peptide microRNAs and hormones, so-called as placentokines, which can be released into the fetal circulation and can be active to develop specific tissues like brown fat and muscle of fetus. To help you guys understand, I would like to explain what is placentokine and what is an exokine. Basically, uh, organs and tissues secrete their unique cytokines and hormone. If it is secreted from placenta, we can call them as placentokine. If it is secreted from muscle, we can call them as a myokine. From adipose tissue, which is an adipokine. And when their secretion is stimulated by exercise, we can call them as exokines. So placentokines, myokines, and adipokines can also be exokines if they are stimulated by exercise. And I'm going to explain the type and role of placentokine in more detail. Uh, First, apelin, one of the placentokines we discovered recently, has function in enhancing mitochondrial biogenesis and thermogenesis in both muscle and brown fat of fetus. Second is the SOD3, which is superoxide dismutase 3, has a role in enhancing hepatic metabolism, glucose tolerance, and insulin sensitivity. Third, adiponectin was also reported as a secreted hormone from placenta. It has been beneficial role in fetal weight management and glucose metabolism. And actually, adiponectin has been originally reported as the most abundant adipose-specific protein and has a role in glucose metabolism and insulin sensitivity. Then placenta also secretes irisin or irisin. This hormone also has function in improving fetal growth and preventing preterm birth. Both apelin and SOD3 have been reported as maternal exercise dependently secreted hormones in the placenta, and these are released into the fetal circulation. On the other hand, adiponectin and irisin have not been reported as exercise-dependent exokines in the placenta yet. But I think considering the important roles of adiponectin and irisin in metabolic health, they may be alternative targets as maternal exercise-dependent placental kinds. So I'm going to explain how Apelin, because we discovered Apelin is one of the placental kind recently, and um, and the metabolic capacity of the offspring in response to maternal exercise, focusing on the apelin. Apelin peptide has a number of benefits in regulating metabolic health. For example, uh, apelin treatment is beneficial in the obese and insulin resistant mice in reducing body weight, adiposity, and triglyceride, and free fatty acid, and um, uh, also beneficial in enhancing insulin sensitivity and glucose metabolism. And our study showed that maternal exercise increases circulating apelin in both mother and fetus. These data showed that potential regulating effects of maternal exercise on metabolic health in both mother and fetus. Indeed, when we started this study, we expected that exercise in pregnancy increases circulating apelin levels in mother, but we did not expect that maternal exercise increases circulating apelin levels in the fetus because blood placenta barrier is existed in the placenta between the maternal circulation and fetal circulation. Thus, we expected that apelin in maternal circulation cannot be released and transferred into the fetal circulation. However, it is also possible if there is a specific transporter for apelin in the placenta, but which has not been reported yet. So there are possibilities that apelin might be released from maternal circulation as hypothesis one, or expressed in the labyrinth zone of the placenta and released into the fetal circulation as hypothesis two, or uh, fetal tissues might secrete apelin itself as hypothesis three. Hypothesis one has not been reported yet, but we could show the hypothesis two and three. Placenta labyrinth uh, expresses apelin in response to exercise during pregnancy, and we can also suggest that fetal brown fat and muscle express apelin by maternal exercise, which uh, I'm going to talk more in the moment. To test whether apelin mimics and imitates the beneficial effects of maternal exercise on fetal brown fat, mitochondrial biogenesis, and thermogenesis, we used apelin intraperitoneal injection model during pregnancy, and which we conducted transcriptome analysis using fetal brown fat, which is same with maternal exercise study, and which shows that apelin during pregnancy enhances mitochondrial inner membrane activity, respiratory chain activity, and brown fat cell differentiation. And these transcriptome data suggest that apelin partially mirrors and partially imitate the beneficial effects of maternal exercise on brown fat, mitochondrial activity, and thermogenesis. Uh, Furthermore, I want to emphasize that apelin treatment during pregnancy also reprograms fetal muscular development, which includes mitochondrial biogenesis and activity, similar with brown fat. And this is transcriptome analysis of the fetal muscle from PBS injection or Apelin injection during pregnancy. The pink is Apelin injection group, and gray is PBS injection group. And this is principal component analysis. And as you can see here, this transcriptome analysis showed that Apelin injection group has the totally different characteristics with PBS injection group. And we can additionally show the unregulation of mitochondrial respiratory activity in the fetal muscle by Apelin administration during pregnancy using transcriptome analysis, which is also similar with the brown fat. And a particular interest for us regarding UCP1 independent mechanisms is calcium fetal cycling in muscle, which is muscle-based non-sugar thermogenesis. And we found that fetal muscle has a unique calcium fetal cycling that involves SERCA and sarcolipin. And in more detail, higher sarcolipin leads to higher cytosolic calcium ion concentration, and these calcium ion can facilitate its uptake into mitochondria, which not only serves as a trigger for oxidative metabolism, but also stimulates uncoupling protein 3, UCP3 dependent thermogenesis in the muscle. So we could suggest that maternal exercise intergenerationally stimulates sarcolipin dependent thermogenesis and UCP3 dependent thermogenesis in the fetal muscle, which is stimulated by Apelin APJ, which is Apelin receptor, and AMPK activation as downstream of G protein coupled receptor. In more detail, we showed that maternal obesity decreases sarcolipin expression and its protein levels in the fetus and offspring muscle. However, interestingly, we could suggest that exercise during pregnancy can prevent the adverse effects against maternal obesity in sarcolipin and its related muscle thermogenesis. And as shown, maternal exercise protected the decrease of surface temperature of early neonates, postnatal, which is the postnatal day 1, caused by maternal obesity, which is also consistent with the sarcolipin gene expression. So then to figure out the mediatory role of apelin in the muscle, we used apelin receptor flux mice crossbreed with beta-actin pre-mice, which is for whole body knockout inducible. And we got the apelin receptor heterozygote knockdown mice because homozygote apelin receptor knocdown mice is has been reported that apelin and apelin receptor are required for placenta development. So most of the homozygote apelin receptor knocdown mice pups were dead. So we used heterozygote model as a knockdown model. Then we could show that the apelin receptor knockdown decreases surface temperature, and the sarcolipin gene and protein expression also consistent. And we also suggested that maternal exercise induced apelin signaling has AMPK dependent downstream signaling pathways. So AMPK knockout mouse model was additionally used to examine the mediator roles of AMPK in sarcolipin and UCP3 dependent calcium ion fetal cycling. And these data show that sarcolipin, UCP3, and phosphorylated CAMKK2, which is calcium conmodulin dependent protein kinase 2, which is a marker for calcium ion activity, and AMPK dependent. All of them are the AMPK dependent. And okay, here's our summary. Started with the fundamental understanding of maternal exercise-induced placenta development, and by understanding of placenta-derived hormones and transcriptome analysis of fetal muscle and brown fat. So now we are able to target significant molecules through the understanding of biological significance of maternal exercise. Especially, we can suggest that intergenerational effects of maternal exercise improve offspring metabolic health, which includes glucose tolerance and insulin sensitivity. Through the activation of non-sugar thermogenesis of brown fat and muscle, with emphasis on the UCP1 dependent and sarcolipin dependent manner. Okay, in my talk, by thanking my previous lab members, particularly most of the research was conducted in the Washington State University in Dr. Du's lab. And I moved to the Baltimore and joined in the University of Maryland School of Medicine last year. So thank you very much for your attention. And if you have any questions, feel free to contact me. And I will leave my conclusion points here in my talk. Thanks again.
Wonderful. Thank you so much, June, for that wonderful presentation. With that, we'll move right on to the Q&A session. And we're going to get started with a first question for you. So the
Okay.
first question I have for you is, could a motorized wheel or one with adjustable resistance be used as a less stressful alternative to a treadmill?
Mm, Actually, I don't know exactly what motorized wheel means, but I think it's going to be like a treadmill exercise. And I have no experience using adjustable resistance on the wheel, but this is an already used method, and I know there is specific equipment in the Columbus instrument as well as mouse exercise model. So I think we can give more accurate answers after measuring the stress levels using that equipment. And I think Chris may have some answers about this. Oh, this is the Chris answers. Yeah.
Okay, great. We'll move on to another question for you here. Val asks if their understanding is correct that only female offspring were protected from diet-induced obesity with maternal exercise. Why did this happen? Were there differences in male and female offspring daily energy expenditure?
Actually, I would like to emphasize that maternal exercise is beneficial in both female and male offspring, but we cannot get any significant differences of the body weight change in the males because when we measure the skeletal muscle mass, um, the male offspring from the exercise mother has a lot of skeletal muscle compared to the offspring from the control mother. So, and when we measure the other metabolic uh, contents, there are significantly there are significant benefits in the males as well. So we can say that maternal exercise is beneficial for both female and male offspring. And yeah, so in our data, most of the data has no significant uh, differences between female and male offspring, even in daily energy expenditure. When we measure the metabolism using metabolic chambers, and which also showed the similar pattern with them.
Okay, perfect. Thank you for that answer. Another question for you. Do you believe that voluntary wheel running versus treadmill running will result in different placental kinds?
Mm, I don't think so because uh, voluntary field running and treadmill running both are the aerobic exercise, and aerobic exercise can secrete the similar placental kinds in my thinking. But if we try to use the resistance exercise in mice, it may be different placental kinds, but clearly voluntary field running and treadmill running has the similar result.
Right, perfect. There's a lot of questions coming through here, so I'm going to just keep rolling through them here. Um, how long do you think metabolic changes in the offspring will last?
Oh, sorry, could you say that again?
Yeah, sure. How long do you think that the metabolic changes in the offspring will last?
Uh, we used 12 weeks of offspring, and that's the final age of age that we measure the metabolic components. And I'm not sure after the 12 weeks later, uh, but when I check the previous paper and previous references, there are a lot of reports that maternal exercise have a beneficial in glucose tolerance and insulin sensitivity, even when the offspring grows up until 52 weeks. But yeah. So yeah, there are long-term effects as well.
Okay, great. I think that answers the question. Another one here from Jacob. Is the Apelin release intensity dependent? Exercise intensity dependent, I'm thinking.
Uh, yeah, it will be possible. It's possible because usually hormones and the peptides can be secreted exercise intensity dependently, but uh, but please, but we need to consider that there is minimal uh, minimal intensity for inducing the hormone secretion. So yeah, I think so. I think so. There will be a possibility.
Perfect. And going down along that line of thought regarding apelin concentrations, do you think changes in synthesis or release drive increase in protein concentration, or could there be changes in clearance or degradation?
Could you say that again?
Sure. So regarding the apelin concentrations, do you think changes in the synthesis or release drive increase in protein concentration, or could there be changes in clearance or degradation?
Actually, I didn't think about that really well, and yeah, I will need to think about more about this, and I may,
Yeah, sure,
uh,
no problem.
yeah,
I'll move on.
answer that question. Yeah.
Okay, cool. Um, Here's a technical question for you. How do you overcome um, in treadmill training when the mice might refuse to run?
Oh, yeah, that's very important and very significant question. So, actually, most of the mice do not want to run. Actually, at the first time they want to run, but after several minutes later, or 10 or 20 minutes later, they do not want to run uh, very well. So, as one of the method we used the spongy as I shown in my slides. Uh, I'm not sure 100%, but the most of the mice do not want to touch with spongy. So when they touch with the spongy, they um, yeah they yeah that's one of the first method. So yeah they yeah we try to uh, use the spongy as an alternative method instead of the electronic shock. So yeah.
Okay, so Jonathan has some questions um, with putting female mice with HFD and getting them pregnant. In his work, they start their diets at 2 months of age, and they wanted to get them pregnant at 3, 4, and 6 months. But sometimes they get pregnant, and sometimes they do not, or they eat their children. How do you achieve it? How could you achieve it?
Oh yeah, we also have the similar experience because obese mother usually it is very difficult to get the pups from the obese mother because yeah we are not sure, but obese mother usually do not care their pups when after exactly after delivery exactly, and that was our challenge as well. So, uh, but when we use the 60% of high fat diet, they usually do not care their pups. But when you try to use the 45% of high fat diet, they can care their pups. So usually we use the 45% of high fat diet.
Perfect. Perfect. Thank you so much for that. Another question here. Um, do you think maternal exercise mimics a hostile ex utero environment that programs the fetus to adopt a thrifty metabolism, so to say, accelerated adipogenesis to ensure survival?
Uh, yeah, maternal exercise. Um, be, yeah. Actually, maternal exercise. The main benefits of maternal exercise is the adaptation. Maternal exercise can adapt the placenta development and can adapt the fetal development. Actually, the acute exercise looks not very good in the specific tissue because maternal exercise, for instance, maternal exercise can uh, enhance the hypoxic condition in the placenta, and we know that hypoxic condition in the placenta is not very good in developing placenta itself and fetal development. So, but the intermittent hypoxia uh, in response to maternal exercise can be beneficial. So, I would like to emphasize that the major important thing is important thing of the maternal exercise is the adaptation. And I think the adaptation can explain your questions as well. So,
Perfect. Thank you very much. Another question here. Are the placental cytokines or hormones that you've mentioned in your talk only generated in pregnant female or non-pregnant female? And do males also generate this kind of bioactive molecule?
yeah, actually, the hormones and peptides I mentioned during my presentation can also be known as myokine or adipokine, which means they can be secreted from muscle or adipose tissue in response to exercise as well. So they can be secreted from tissues, including the placenta, uh, uh, even non-pregnant women or pregnant women as well from the muscle and the adipose tissue. So, but what I'm talking is uh, they can be secreted very well during the exercise, and these secreted in response to exercise can be beneficial to reprogram the placenta and fetus.
Right. And so going along with these bioactive molecules, how do you discriminate between them? If you're assuming that they're released into the circulation and that they're not tissue specific, um, they're not dependent on tissue, how do you discriminate between them?
Oh, actually, yeah, for instance, apelin in the circulating level, it can be secreted from the muscle or adipose tissue or the placenta as well, but we do not measure and we do not experiment any experiment uh, for for examining these apelin released from the specific tissues. But yeah, this will be conducted in the future. So
Excellent. Okay. I think I have time for one or two more questions here, but in your opinion, out of the three thermogenic mechanisms promoted through maternal exercise, SLN, the UCP1, and BAT, which one do you think is the most important?
um, I can't say that because the UCP1 is traditionally known as a number one thermogenic pathway, but the other signaling the other one like UCP3 or sarcolipin and the SARCA signaling in the beige fat, I think the most of the signaling is very important because in the biological systems, um, there are a lot of and bunch of the signaling pathway is very important, uh, and the UCP1 and sarcolipin and UCP3 can be active in the different tissues, and I think the most of the signaling you mentioned is very important. So I can't say uh, the number, the number one. So
Yeah, of course. Um, one last question I'll push for you. From a drug metabolism and pharmacokinetic perspective, have
mhm.
the activities of drug clearing enzymes been evaluated in the pups to understand if there are potential CYP expression differences?
um, mm, actually, I didn't think about this very well, so I need to think about it more. Yeah, but in the drug discovery, uh, the actually I cannot answer very well in the questions, but I would like to explain the the initial reason why I focused on the exercise study, because exercise can stimulate a lot of hormones and a lot of cytokines, and these hormones can be active very well. And I think if we find and if we target the specific molecules and specific peptides uh, uh, as pharmaceutical targets, I think it can be to uh, useful in the future. So, but I think this is the very initial stage to discover and to target and to find some drugs for them. Yeah, but yeah, this is very important, and I think it can be linked to the translational research as well.
Okay, great. Well, thank you so much, June, for your presentation today and for the answers to all these questions. We really do appreciate your time talking to us all today.
Thank you very much.
In closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day.